So perhaps when you think about genomics and ART, the first thing that comes to mind naturally is PGT. Make no mistake about it, successful identification of euploid embryos absolutely is critical to IVF success. And this is also an area that CooperSurgical spends significant effort innovating and developing out solutions. Our PGT-AI platform is summarized here, and you can find more information at our website or our YouTube channel. But for the purpose of today's talk, we're going to be starting with the assumption that we've identified euploid embryos and then discussing additional factors that influence IVF success. Initiation of a successful pregnancy really begins with implantation, which is a remarkable and complex process. From the embryo perspective, we start off with a hatching embryo, which becomes juxtaposed to the lining of the endometrium. There are various adhesion and signaling molecules involved, which we'll talk about in a bit. And then the embryo becomes attached and adheres to the lining of the endometrium, where it then proceeds to invade. Speaking of the endometrium, it's defined as the inner lining of the uterus, which is actually a pretty interesting and unique organ because of the dynamic changes it undergoes every 4 weeks, known as the menstrual cycle. Histologically speaking, the menstrual cycle can be characterized by some very distinct differences. Starting in the proliferative phase, we have these tight, compact glands. Progressing through, we see an expansion of those glands and the luminary epithelium. And then in the secretory phase, we can see that those glands are in fact filled with secretions. In the absence of an implanting embryo, we then, of course, proceed to menstruation. Now, while I've presented the embryo and the endometrium as distinct entities, it's important to remember that these complex processes must be occurring in sync with one another. And so on the left, we see the complex processes of fertilization through cleavage stage development into maturation of advanced blastocysts, while at the same time, we know that the endometrium is undergoing both proliferation and differentiation as it prepares to support the implanting embryo. Anytime synchrony is of concern, of course, the timing of the underlying components is important. When we look at the embryo, we know that some of the early divisions are driven predominantly by maternal mRNAs, and it's only until late cleavage stage that we begin to see the embryo activate its own genome and start reacting to some of its own signals. On the endometrial side, it's driven, of course, by the steroid hormones produced predominantly by the ovaries. So in the early stages, while follicles are developing, we see significant estrogen activity shown in blue. Following the LH surge, we get ovulation and differentiation of the underlying follicle into the corpus luteum, and then we begin to see progesterone-mediated physiology. Again, the key takeaways here are that we have complex physiological processes occurring simultaneously in both the embryo and the endometrium, driven by complex molecular regulators. Many of the molecular regulators involved in these processes have been well defined, and some of them are listed here. I don't want to focus in on any of them in particular other than to again demonstrate that there are multiple players involved here and it's quite complex. But these molecules work in orchestration to drive multiple processes. On the embryo side, on the left, really the key pathways in physiology being driven are embryonic growth and survival. And then in the middle, we have the endometrium, where we're driving proliferation to produce more cells to support the embryo and differentiation to secrete and produce the products necessary for embryo development. And then finally, we do know that there's significant signaling and interaction between both the embryo and the endometrium. And so both the seed and the soil are driving the interaction here. Some of the molecular networks at play during early pregnancy function as critical regulators of communication, of which there are multiple modalities. On the left-hand side, we see paracrine signaling. This can be regulated by cytokines and morphogens, for example. And here we have signaling of adjacent cell types that are nearby to one another. In the middle, we have autocrine signaling, where we look at, again, things like cytokines and growth factors, in which a given cell type signals very close by to other cell types of its same kind. Finally, we have endocrine signaling, and this is really where a certain cell type may produce molecules that circulate through certain systems like the blood system to distal cell types and have an influence there. Considering the signaling paradigm of the peri-implantation period, we of course have endocrine signaling where the steroid hormones are signaling to the endometrium, but specific to the implantation process, we have autocrine signaling in which signals derived from the embryo signal back to the embryo itself. And then we have paracrine signaling, in which communication from the underlying endometrium provides signals and cues that are necessary to support development of the embryo. This dialogue becomes of extra importance when we consider culturing embryos in vitro. Autocrine signaling is one of the hypotheses why some favor group culture, because embryos in a single droplet may have opportunities to signal to one another and provide some of those important cues. However, of course, absent is the signals derived from the underlying endometrium. And therefore, it's intuitive that if we can identify what those critical signals are and potentially supplement our culture media with those signals, we might be able to improve embryo development. One particular class of signaling molecules are cytokines. Cytokines are secreted proteins that have a receptor on their target cell where they exert their effects. Cytokines are probably best known for their role in inflammation, but they can exhibit really a whole range of responses on their target cell population. And this can be driven by both the dosage of cytokine present and the mix of various cytokines present. Importantly, cytokines have been shown to play a role in many of the processes during early pregnancy. I won't go through all of them listed here, but of course, of particular relevance are the impact on blastocyst viability and survival. One particular cytokine that we've been interested in for a few years now is GM-CSF. This cytokine has been well studied, and as we reviewed some of the evidence in literature, a few things stuck out to us. Interestingly, we noted that it can be detected not only in the fallopian tube, but also in the uterine epithelium, and it's abundant in uterine fluid. Next, we noted that its peak concentrations are detected during the mid-luteal phase, which is right when we would expect implantation to occur. Moreover, we noted that GM-CSF levels are reduced in women who suffer from recurrent pregnancy loss. And then perhaps most intriguingly, we noted that GM-CSF is not produced by the embryos themselves. Honing in on the role of GM-CSF in embryo development, we have three really interesting studies here. In each, we're looking at a scenario in which embryos were grown in IVF media with and without GM-CSF. On the left, we have data looking at the impact of supplementation on embryo survival. What we can see quite strikingly is that there are significant increases in survival, especially beginning at the morula stage, in which embryos grown in GM-CSF survive at a better rate than those without. The next two graphs get at some of the underlying mechanisms that may be contributing to this survival. In the middle, we have glucose uptake, and what we see is that the embryos supplemented with GM-CSF have an increased uptake in glucose, meaning that they're more metabolically active. On the right side, here we are looking at apoptosis or programmed cell death. And again, what we notice is that embryos supplemented in GM-CSF displayed decreased cell death. And this is true not only in the inner cell mass, but when we quantify all cells. Moving from the culture dish to clinical outcomes, here we have three different studies that show more or less the same thing, but I wanted to include all three of them to really underscore the fact that this is a result that's been reproduced and seen in multiple populations. In each of these three studies, we're essentially comparing embryos that were cultured, again, with and without GM-CSF. Rather than going through every study and every category, you can simply note that the categories which achieve statistical significance are labeled above. Summarizing some of the findings, we see that while the second study does trend towards an increase in implantation, clearly in the first study we didn't see an increase in positive HCG. But interestingly, it seems that supplementation with GM-CSF is producing healthier embryos that instead are able to survive better. We see increased ongoing clinical pregnancies, increases in live birth rates, and importantly, of course, decreases in miscarriage or in loss rates. Of particular interest is on the far right, where we look at patients known to suffer from recurrent pregnancy loss. And here we can really see that the differences are quite striking. Continuing to explore the role of GM-CSF in poor prognosis patients, as indicated by the graph on the right. On the next slide, I'm going to take the study on the left and we'll separate out the poor prognosis patients. So the data on the left side, again, is the same as the previous slide. However, on the right side, from that population, if you isolate the patients who suffer from recurrent pregnancy loss, what we find is that the difference between patients who had embryos grown and supplemented in GM-CSF versus those that did not is more striking when we look at ongoing pregnancy either at 7 or 12 weeks, and then again at live birth outcomes. Together, these data indicate that GM-CSF can result in healthier embryos that not only survive longer in pregnancy, but these effects are exaggerated in our poor prognosis patients. Recapping some of the discussions we've had today on GM-CSF, it's quite evident that it plays a critical role in embryonic development. Here we've included a table that highlights some of the physiological effects of GM-CSF on embryo development. We didn't cover all of the effects, although many of the studies that we highlighted did go into some further detail here. In sum, what we've observed is that GM-CSF promotes progression from early zygote stage to blastocyst. It does this by normalizing cellularity via a couple different mechanisms, including increasing glucose uptake to drive growth and decreasing stress response and apoptosis to promote cell survival. Together, these impacts synergize to increase the developmental competence. As we try to translate that to the clinic, what we observe is that there's increased rates of ongoing pregnancy as well as live birth rates. Importantly, these effects are even more significant and more marked in our poor prognosis patients that we struggle to get pregnant, and the patients who we've seen have one or more miscarriages. For all of these reasons, CooperSurgical is proud to supply IVF culture media supplemented with GM-CSF. We actually offer a couple different choices here depending on whether you prefer a single-step media or sequential medias. In the single-step media, we offer our SAGE one-step supplement with GM-CSF, and for the sequential IVF culture medias, we offer our EmbryoGen and BlastGen product lines. If you recall, we began discussing GM-CSF under the context of supplementation. The reason why GM-CSF is a good candidate for supplementation is because we discussed how embryos don't produce the cytokine themselves, but rather it's produced by the endometrium during the peri-implantation window. So while the first portions of the talk really discussed the role of paracrine signaling and the impact of embryo development, I'd like to shift gears now and talk about development and synchrony of the complex physiological changes that occur in the underlying endometrium. As a quick refresher, the endometrium is the inner lining of the mammalian uterus, which is a dynamic organ that undergoes cyclical changes as part of the menstrual cycle. First, we're driven predominantly by estrogen, which drives proliferation of the luminal epithelium and endometrial glands. And then following ovulation, really we shift to more of a progesterone-mediated biology. Now, in contrast to some other animals in which the embryo is really the key driver of pregnancy outcomes, the endometrium plays a really important role in human reproduction. The endometrium actually has remarkably sensitive ways of acting almost as a sensor to evaluate the putative implanting embryo. If everything goes well, implantation triggers a proliferative and differentiation response of the endometrium known as decidualization, which nurtures and supports the developing embryo and eventually contributes to the placenta. However, if the developmental competency or viability of the embryo is lacking, the endometrium can sense this and it will trigger menstruation so that it can begin moving on to the next cycle. In case some of that discussion was a bit abstract, here we have a nice visualization of some cellular compartments and molecular mechanisms involved during the implantation period. Going across the top of the graphic, you see LE standing for luminal epithelium, and then the invaginations where you'll find GE signaling glandular epithelium. While the luminal epithelium is a site of attachment and the initiation of implantation, the underlying stroma plays a key role in the decidualization process necessary for continued implantation. Stromal cells, though not many pictured here, are the blue spindle-shaped cells adjacent to the epithelium. As we discussed earlier, there are complex molecular networks and mechanisms that drive physiology. There's a lot of them listed here, and of course we're not going to go through all of them, but I do want to hone in and reiterate the role of the steroid hormones estrogen and progesterone. Though many in IVF and ART are very familiar with these steroid hormones, recall as a refresher from your molecular biology days that these hormones work by binding their cognate receptors, the estrogen receptor and the progesterone receptor. And these molecules actually serve as transcription factors, shown here for estrogen receptor and progesterone receptor in an epithelial cell, or estrogen receptor and progesterone receptor in a stromal cell. Transcription factors are key to physiology because they regulate the transcription of many of our genes. Again, we won't go through all of the gene networks demonstrated here, but the key takeaway is that the endometrium is a hormone-responsive organ. The hormones estrogen and progesterone can act in both the epithelial and stromal compartments, where they can have different, sometimes synergistic, sometimes antagonistic interactions to drive gene networks. And these gene networks can then be used to get information or to indicate the physiological status of the endometrium, which will be important later. Taking a step back from some of the complex molecular networks, the ultimate goal and objective of the endometrium is to support an implanting embryo. As we discussed earlier, the endometrium is a very dynamic organ and it's constantly undergoing changes. In fact, throughout the 4-week menstrual cycle, there are really only a few days in which the endometrium is receptive or willing to receive an implanting embryo, and this phase is known as the window of implantation. In natural cycles, the window of implantation occurs approximately 7 days after the LH surge, in most women, that is. In hormone replacement or controlled cycles, typically progesterone is administered, and the idealized window of receptivity is considered to be 5 days after progesterone administration. The concept of the window of implantation, or restricted receptive time, really originated and was first hypothesized in the 1950s when endometrial dating really first came to be based on histological examination, such as I showed earlier in the presentation. The presence of a narrow receptive window surrounded by refractory periods later was shown to be clinically relevant in the 1990s, and for decades it was thought that all women would have similar underlying physiology. As we all know now, following the successful Human Genome Project, science saw a tremendous explosion in capabilities. Many tools were subsequently developed, and with those tools we began to really gain an understanding of the molecular mediators of our physiology. With that increased understanding, we began to define many of the molecular networks and interactions that I've described previously. Scientists then began to use these newly developed tools to investigate some of the differences in our physiology and the molecular mediators in various different conditions. Without any surprise, the uterus, such a dynamic organ, really offered a lot to discover. Starting broad and drilling down, scientists first really focused in on molecular differences and changes in the endometrium throughout the menstrual cycle. In the graphic here, that's demonstrated on the x-axis of time. So starting on the left, we have a dotted line signifying tissue that could potentially represent menstrual tissue, and we continue to the right to have early proliferative, late proliferative, early secretory, mid-secretory, and late secretory, including the window of implantation right at the beginning of mid-secretory phase. Moreover, as our tools got better, we were able to begin characterizing and profiling various different cell types. So in this graphic, you can see multiple cell types with a key on the right, and on the top and the bottom of the figure, what we're actually showing here is gene expression profiles of several samples of epithelial cells and stromal cells throughout these cycles. Now, we won't key in on what the specifics are here, but each vertical little stripe is a single gene. And as you can see how that gene, based on the color intensity with the code on the bottom left, changes in the respective compartments and in the respective menstrual phase. Though you may not be able to make perfect sense of it, hopefully your eyes can pick out some patterns and some differences throughout the illustration. Eventually, what scientists were able to do was to leverage statistical classifiers and various different algorithms to establish a gene signature that reflected any given cell type or any given phase of the menstrual cycle. Once these signatures were established, we could then begin comparing patients who presented different phenotypes and prognoses and compare their signatures to one another. Using the previously discussed techniques and signatures is when we eventually came to discover that not all patients are the same. And so in the middle, we have the typical scenario, if you recall, 5 days after progesterone administration or 7 days after the LH peak. We would expect the endometrium to be receptive as well as to have a receptive signature. If we were to examine the signature of that patient earlier, say 2 or 3 days after progesterone administration, or perhaps later, say 7 days after progesterone administration, we would expect to find signatures that perhaps reflected more the early secretory phase or the late secretory phase, respectively. However, in some patients, we didn't always see this. And so on the top, you have what we call as pre-receptive or delayed window. And in this case, if you perform the biopsy at 5 days after progesterone administration, when you expected to see a receptive signature, you would actually see a signature that indicated you hadn't reached the receptive window yet. And in fact, if you biopsied a subsequent day, you would find a receptive signature later than you expected to find. In the exact opposite scenario, known as post-receptive, if you biopsy that patient at 5 days after progesterone administration, you would find, in fact, that the gene signature had advanced and had passed the window of receptivity. In patients with these kinetics and physiology, you would actually have to perform a biopsy at an earlier time point to identify a signature that suggests that the endometrium was receptive. Here we demonstrate the translation of these gene signatures to clinical practice. So again, recall we may have a few patients, some who are receptive, pre-receptive, or post-receptive. When we conduct molecular analysis, as demonstrated here below, we can look at the gene expression and establish gene signatures that reflect those physiological states. These signatures can be transformed, statistically speaking, into a two-dimensional space where we see unique populations. In green here, we have a group of patients who are populating close together that we know to be pre-receptive. In purple in the middle, again, we can see a distribution of patients who are demonstrating a receptive signature. And then over here, we have a group of patients who are demonstrating post-receptive gene signatures, and then finally a fourth class of non-receptive signatures. When we look at the gene signature derived from patient 1, we see that this sample clusters right in the middle of the pre-receptive group, and therefore this patient must be pre-receptive. In contrast, when we look at patient 2, we see it mapping and identifying in the post-receptive group, and finally here patient 3 is mapping in the middle of the receptive group. If you'll recall from the previous slide, in addition to using the receptivity terminology, we also discussed delayed and advanced windows. And so, of course, patient 3 is our easiest patient who has a receptive window, which means that her endometrium is prepared to receive the embryo at exactly the time that we would expect, and transfer should be performed at that same time. In contrast, where things get a little bit more difficult, we have patient 1 who is pre-receptive or has a delayed window. This means that as far as physiology and gene signatures indicate, her endometrium hasn't advanced to the window of implantation yet, and therefore transfer would be better suited if it were delayed. Finally, we have patient 2 who is post-receptive or has an advanced window. In this case, the gene signature indicates that her window has already passed, and the ideal time for a precision transfer would be 1 day earlier. Based on these principles, CooperSurgical has identified gene signatures that reflect the various receptivity states, and we are proud to present ER-peak endometrial receptivity testing. This test is intended to be performed on endometrial biopsies that were obtained under the same timing and hormone paradigm that you would utilize for embryo transfer. While some of the concepts related to receptivity, gene signatures, molecular mediators, etc. are pretty complicated, we're happy to present the results in a simple and easy to understand graphic shown here. If the patient tests receptive, then we've identified the ideal time to transfer an embryo under the exact same conditions in a subsequent cycle. If that is unfortunately not the case, the subsequent cycle, the embryo can be transferred with a shifted window, either 1 day early or 1 day late, or a second biopsy can be taken for confirmatory testing to confirm that the shifted time period reflects a receptive status. So far, causes of a shifted window still need to be investigated. However, we do know that patients with recurrent implantation failure, as well as patients with higher BMI, exhibit an increased incidence in a shifted window. These patients are often considered ideal candidates for receptivity testing. Additionally, some patients who have only a single embryo available or a single euploid embryo available also may find receptivity and window testing very important. In approximately 2 years of testing, CooperSurgical has observed that approximately 60%, the majority of patients, are in fact receptive. However, approximately 2 in 5 patients presenting and requesting ER-peak testing are in fact not in the receptive window. Of those patients, we do see approximately 27% of them who are pre-receptive, while approximately 7% of them display post-receptive signatures. It's important to note that within these data, we have not stratified out any patients based on their indication. Identifying altered physiology is one thing, it's great, but what about implementing intervention and clinical practice? Here we have clinical data from 2 very compelling and interesting stories. On the left, we have patients who, in a retrospective study, were identified to have a shifted window. In the dark blue bars, this was their first cycle under the standard time frame, and we can see relatively poor outcomes. Once the shifted window was discovered, a subsequent embryo was transferred in the precision embryo transfer period. In other words, in accordance to the signature observed, and we can see marked improvements in implantation rate, clinical pregnancy rates, and ongoing pregnancy rates. It should be noted that in the original standard embryo transfer, not all pregnancies were followed to full ongoing pregnancy, and that's why that data is missing. The study on the right is separate patients in contrast to the patient-controlled study on the left. And in this population, we have recurrent implantation failure patients who have undergone standard embryo transfer during typical timeframes, or those who have had receptivity testing and then undergone a precision embryo transfer. And again, we see significant increases in implantation rate, ongoing pregnancy rates, and live birth rates. So to summarize today, we discussed both the seed and the soil and their critical role in establishing a pregnancy and implantation. Supporting embryo development and embryo culture, we discussed the roles of the cytokine GM-CSF in promoting growth and survival and development of embryos. Those viable embryos are precious and rare, and we certainly want to ensure that we're giving them the best chance to implant. And so then we discussed how steroid hormones regulate the cyclical changes of the endometrium, and how gene expression and gene signature testing via assays like ER-peak endometrial receptivity testing can help identify the optimal time to replace that precious embryo and achieve the best pregnancy outcomes. So with that, I thank you for your attention today.